In the next 3 chapters, we'll be examining the differences between the bacteria and archaeans, then the eukaryotes, and then we'll take a look at viruses. In this chapter, we'll be looking at prokaryotes, predominantly the bacteria, and then we'll make some comparisons between bacteria and archaea towards the end of the chapter. Remember, the prokaryotes have been around for 3.8 billion years, so they're really highly adapted to the spaces in which they live, even more highly adapted than we think we might be. If you think about it, they've had 3.8 billion years to get good at it. So first of all, let's review how we define what is prokaryotic. We used to say a lot about what they're not, but it's more important to think about what they really are. There are 3 distinct ways that we'll consider what they are. The way that their DNA is packaged. For example, a prokaryotic cell has DNA that's not packaged in a nucleus. They might have 1 or a few circular chromosomes, but not nearly as many as we see in a eukaryote. We look at the makeup of their cell walls. Do they have a unique cell wall structure? For example, bacteria have peptidoglycan as their cell wall. And archaea have another tough material that's very distinct from that of bacteria. However, both have cell walls, and eukaryotic cells do not. Finally, we look at the internal structures. The internal structures in prokaryotes are much less complex. They're not membrane bound as they are in eukaryotes. And part of the reason for this that we'll consider is the size. The surface area to volume ratio is what's really key here. Essentially, because there's more surface area to the volume ratio, that means there's more surface area accessible to every single point within the cell. So it's easier to deliver stuff to the cell surface and receive stuff from the cell surface and bring it into the center of the cell. So they really don't have a need for all this complex internal structure. Let's look at the dimensions as an example. If we look at a prokaryote with an average of 1 micrometer and a eukaryote with an average of 30 micrometer, we'll see that that's about 30 times smaller. We'll see that the prokaryotic surface area is 900 times smaller. When we consider the volume, it's 27,000 times smaller in a prokaryote than it is a eukaryote. So that allows us to have a surface area to volume ratio, which is 30 times larger in the prokaryote than it is a eukaryote. This surface area to volume relationship affects how an organism transports material and accomplishes its metabolism, which is often defined as getting rid of heat or wastes. The bottom line is in a prokaryote, everything is close to the surface, so they don't need all the complexity of structures. But let's take a look at what they do have. When we consider a prokaryotic cell, we'll look at the general organization of the cells. We'll be considering the external structures. Then we'll take a look at the cell envelope in lots of detail, which includes the outer membrane as well as the cell wall. And then the internal structures we'll observe. We'll see cytoplasm, ribosomes, other cellular inclusions, the nucleoid or chromosome region of the cell, as well as the cytoskeletal elements, and the endospore formation. So this is a basic outline of where we're going in the next few sections of this chapter. So here are some structures that are common to bacterial cells. All bacterial cells, as we've already established, have a cell membrane, they have cytoplasm, ribosomes, and 1 circular chromosome. Sometimes they have a couple of extra small accessory chromosomes, but we'll look at those in more detail soon. Most bacterial cells have a cell wall, as well as a surface coating or glycocalyx, which we'll look at in much more detail. Some other structures that we might find in bacterial cells are flagella, or a tail-like structure for swimming, pili, or channels for communication between the cells, fimbriae, little motile projections off the side of the cells. They may have a capsule around the outside of the cell, some slime layers, some other inclusions inside the cell, an actin cytoskeleton to keep the cell in a certain structural shape, and then endospores that are used for reproduction. Again, we'll be looking at all of these in more detail as we progress through this chapter. In this chapter, again, we'll be focusing on the bacteria. We'll make some comparisons to these bacteria later in the chapter when we look at some distinct features that we find in archaeans. As you've probably established by now, looking in a microscope at a two-dimensional prokaryote is sort of difficult because they appear fairly featureless. It's not until we get the details that we're able to see through electron microscopy and then other details we've learned through biochemical studies that we get a nice three-dimensional shape. Here in this figure, we've presented you with a nice three-dimensional shape, and we'll look at all these organelles in a little bit of detail right here. First of all, let's look at the glycocalyx. The glycocalyx, it's a layer of molecules or a coating around the cell, external to the cell wall. It serves protective, adhesive, and receptor functions, and it may fit tightly, or it could be very loose and diffuse. Next, the bacterial chromosome, or nucleoid, is located inside the cell. It's composed of condensed DNA molecules. DNA is going to direct all of the genetics and heredity of the cell and codes for all the proteins that it needs throughout its life. Often we'll see a plasmid, which is a double-stranded DNA circle that contains some extra genes. The pilus that you can see here is an elongated hollow appendage. It's used for transfer of DNA to other cells, exchanging genetic information in reproduction. So it's an appendage used for reproduction. A prokaryotic cell also has ribosomes. As we've already discussed, those ribosomes are different in structure than the eukaryotic ribosomes. These are composed of proteins and DNA also, and they're the site for protein synthesis, so their function is the same, although their structure is different. The cytoskeleton, again, is made of actin predominantly. It's composed of long fibers of proteins that encircle the cell just inside the cell membrane and help keep its structure. You can think of it much like the framing of a house. Now, the flagellum is a specialized appendage in filament form that rotates, and this rotating movement pushes the cell along through its environment. There are fimbriae, which are fine hair-like bristles. These bristles extend from the cell surface and help it adhere to other cells and surfaces. Next, we'll look at inclusions. Inclusions are granules. They store nutrients such as fat and phosphate or glycogen so that the cell can have access to these energy stores whenever it needs. The cell wall, again, is a distinct feature of prokaryotes and eukaryotes. It's a semi-rigid casing that provides structural support and shape for the cell, so it works in conjunction with the actin cytoskeleton. Right beneath the cell wall is a cytoplasmic membrane, which is a thin sheet of lipid and protein, very similar to what we see in eukaryotes. It surrounds the cytoplasm and controls the flow of materials into and out of the cell. Some cells have an extra membrane that's similar to the cell membrane and also contains lipopolysaccharide. This outer membrane controls the flow of materials, and then portions of it are toxic to mammals when it's released. The endospore, which is not shown in this picture, is a dormant body formed within some bacteria that allow for them to survive adverse conditions for long periods of time. Finally, the cytoplasm, just as in a eukaryotic cell, is a water-based solution filling the entire cell. In the rest of the chapter, we'll look at each of these structures in more detail as we move from the outside to the inside of the cell.